This is the first part, or part A, of a multi-part lecture series on the fundamentals of microbial growth. Once you've completed all of the parts of this lecture, you should be able to understand how bacteria and archaea use binary fission to replicate a cell, know what proteins are relevant to binary fission and those that are targeted by penicillin, understand the concept of microbial population growth and be able to describe a microbial growth curve, know how to apply the concept of generation time and perform basic calculations that allow you to predict bacterial numbers, know how microbes are described by their temperature preferences and what the molecular adaptations they have that allow them to grow at very high or very low temperature, know how organisms are described with regards to their oxygen use and tolerance, and appreciate the enzymology important in managing oxygen stress, and understand how microbes manage osmotic stress and how they are described with regards to their salt tolerance. All of that might sound a little bit dry, but it really is the underlying biology that explains some of the amazing diversity of microbial life on Earth. And it'll help you answer some fun questions, I think, like why is it always fungus growing on top of my jam and never bacteria, if you've ever wondered that, or why it's usually fungus growing in your coffee grounds and not bacteria, or why did the ancient Egyptians use honey in embalming? Very costly fluid, why would you use it in embalming? And why do you almost never find anything growing in your maple syrup, even if you don't keep it in the fridge as per recommendations? So the first part of this lecture is going to address cell division, the process of binary fission, and it's going to talk and address the proteins and structures that are part of the cell machinery responsible for cell division, which is referred to as the divisome. So microbes reproduce by binary fission, and specifically I mean the microbes that are in the bacteria and archaea, those two domains. There are some other options besides binary fission, but we're not going to cover them in this class. Before we begin, let's define microbial growth and define it as an increase in cell number. That's a little bit different than thinking about the growth of a single human being, where you would both have increase in cell number, but that would contribute to the growth of only one single organism. In the microbial world, growth is defined as an increase in cell number. In order to get an increase in cell number, you have to double the cell, one single cell, and we're going to talk about that process, binary fission. So binary fission may initially seem very simple to you, and it perhaps is when you compare it with the complexity of mitosis and cytokinesis that occurs in a eukaryotic cell, but in fact binary fission is a very complex, highly orchestrated process facilitated by a suite of proteins and specific organic molecules. It's still a topic that is actively researched, and we still don't understand how a lot of different kinds of bacteria and archaea actually do binary fission. The cell machinery that accomplishes binary fission is referred to as the divisome. So the basics of what happens during binary fission are shown here in this figure, but it's missing, a lot of crucial details. And so I want you to be remembering as you look at this, this figure that in order to replicate the cell, you have to first replicate all of the internal contents of the cell. So it's really important to remember that the chromosome, ribosomes, the cytoplasm, the cytoplasmic membrane, and other contents all have to be replicated, and they have to be split evenly between the two cells. The evenness of that division results in a growth pattern that's referred to as balanced growth. In other words, the two daughter cells are identical. I also want you to realize that it's very, very important that the chromosome as it forms, so we go from one circular chromosome to two chromosomes, and then they have to be pulled apart so that they each make it to the new daughter cells. And they will do that by being attached to the cytoplasmic membrane on either side, and the active cell division will pull them apart. But it's very, very important that each chromosome makes it all the way into its appropriate location, and that you don't end up with two daughter cells where one has an incomplete chromosome. And the divisome includes proteins whose job it is to ensure that the timing of separation is correct. The septum refers to a cross wall that forms between the dividing cells. In rod-shaped bacteria, that will happen exactly dead center, and the same is true with coccus-shaped bacteria. There's some variation in the natural world that I won't get into where there are some examples where that doesn't happen, but we'll just focus on exact cell division. The septum, a cross wall, is what forms here. The time that it requires for one cell to double into two is referred to as generation time, or G. So G is the time required for one cell to double or replicate. It also means that G is the same time required for two cells to become four, or for four cells to become 8. With each generation, you will have a doubling of both cell number and also mass. So if we look at this table, this illustrates some of the known doubling times or generation times for a variety of organisms: bacteria, and then algae, protozoa, and fungi. The algae, protozoa, and fungi are all eukaryotes, of course. You should know that. Okay. And one of the patterns you'll notice here is that they tend to have longer generation times than the bacteria. There are some exceptions to that. For example, Saccharomyces cerevisiae down here. That's what we use for making beer, wine, bread, and other things. It has a really rapid doubling time that's comparable to some of the times seen in the bacterial world, but in general, eukaryotes take longer. Bacteria are faster. It's an advantage that they have over eukaryotic cells. You'll also notice that a generation time is provided for each species given. It is a quality representative of the species, but however, it is affected by conditions. So things like the temperature or the nutrient availability will affect generation times. Take a look at the bacterial information. Let's compare E. coli over here, which has a generation time of 0.35, so that's a decimal hour. That's, roughly 20 minutes, okay? And let's compare it with Mycobacterium tuberculosis, causative agent of TB. It's got a generation time of around 12 hours. That's a pretty big difference. So in one day's time, you would expect to have only 2 generations of Mycobacterium tuberculosis. That's a very slow-growing bacterium. E. coli, in contrast, is quite rapid. There are known bacteria that are halophiles that have generation times of 7 to 10 minutes, and that's probably the fastest or shortest generation time known. Why would anybody care about this? Well, it's got all kinds of relevance to humans. So if you're trying to predict how quickly a known pathogen could grow in your body or in some food that you might have out on the counter, then if you want to be able to predict that, you need to know the generation time for that particular organism and what it is under those particular conditions. So we study generation times so that we can better control microbes, and also so we can so that we can control them and limit their growth, but also so that we can control them and make predictions when we're using them in industry in order to make things like alcohol or bread or food products or pharmaceuticals. So there are all sorts of people that study these sorts of things. So the divisome is the cell machinery that accomplishes binary fission. Proteins called FTS proteins assemble in a ring that is precisely around the center of the cell, assuming structures as shown in this figure. Each of those structures is a protein or complex of proteins that plays a role in the divisome. FTSZ proteins form a ring around the cell center. They're shown there in yellow. So that's these guys here. Okay? So they form a ring around the cell center, and this becomes the cell division plane. It's going to form exactly at midpoint. FTSZ is a protein that is functionally similar to tubulin. It's not similar in its amino acid sequence, but it does the same function basically as tubulin, a protein found in eukaryotes. One of its roles is to catalyze the hydrolysis of GTP. That's shown in this figure here. So that's an energy releasing reaction. This is where the energy for the polymerization of FTSZ comes from. Each of these little balls is a single FTSZ protein. They polymerize together to form that ring structure. FTSA and ZipA, here's ZipA and here's FTSA. FTSA and ZipA anchor the FTSZ ring to the cytoplasmic membrane. FTSI, which is shown here, and I've mentioned this before in class, FTSI is a peptidoglycan biosynthesis protein. FTSI is also one of the known penicillin-binding proteins in the cell. FTSK plays a role in helping the chromosome to separate. FTSA, shown here, is also, it plays a role in cell division, and one of those roles is that it is an ATPase. That means that it can hydrolyze ATP, yielding ADP and a phosphate group. That's an energy yielding process, and it provides some of the energy required for the divisome to do its work, which is an overall energy requiring process because it is a synthesis process. So this is happening, so just to make a stronger point about that, GTP and ATP hydrolysis are both involved in providing the energy for cell division. It's important, as I mentioned before, that the replicated chromosome, which produces 2 copies of the chromosome, that each of those copies makes it to the opposite pole of the cell prior to formation of the septum and cell division, so that each daughter cell is intact and has a complete chromosome. So what will happen is DNA replication will occur, forming 2 identical chromosomes. These are each attached to the cytoplasmic membrane. It's really important that each chromosome makes it to a pole of the cell, and what facilitates that process is the coordinated activities of proteins known as MIN proteins. MIN-CD is shown here, okay? And you'll note that it will organize in this kind of spiral pattern around the cell in the cytoplasm, shown in this figure here, for example, there, and there, okay? The MIN proteins are going to coordinate with the FTSZ proteins and execute the timing of the formation of the FTSZ ring very, very precisely. So as the cells begin elongating, the MIN proteins work together in the cell to control the timing of the formation of the FTSZ ring, allowing sufficient time to elapse for the elongating cell to pull the 2 chromosomes apart and keeping the formation of a septum at the exact midpoint of the elongating cell and nowhere else. So the MIN proteins coordinate with the FTSZ proteins so that the septum forms at midpoint, okay, exactly halfway, and so that the septum does not form prematurely, ensuring that the chromosomes make it into the new daughter cells. The proteins of the divisome coordinate the timing and location of cell division, but they also play a role, at least in some cases, in the final shape or morphology of the prokaryotic cell to which they belong. So we know that there's a very important protein called MREB, for example, and as well as that, some actin-like proteins. These are proteins that have homologs to eukaryotic cytoskeleton proteins, and these are the proteins that make up the prokaryotic cytoskeleton. So bacteria and archaea do in fact have cytoskeletons, just like eukaryotic cells. Interestingly, if you mutate the protein MREB so that it is no longer functional, the cell in which you have done this will default to a coccus shape. So if you start with a rod-shaped cell, mutate its MREB, the result will be cocci-shaped cells. In other words, a morphological transformation will occur. And so we think some of the people who study the evolution of microbial cells think that the coccus-shaped cells may actually be the default state or the original state, and that things like rods and spirochetes evolved later. In any case, it is clear that MREB does control or influence cell shape. In the cell, it'll form spiral bands, and that's what's shown here in purple, oscillating around the cell. and those bands will be attached to the cytoplasmic membrane and the peptidoglycan. They will be somewhat mobile within the cytoplasm despite those points of attachment, and they appear to be constantly moving as cell division occurs, and that seems to be what is controlling the morphology, the final morphology of cell shape or cell shape. So in order to have cell division, you have to make new cell walls. And in order to make new cell walls, you have to make new peptidoglycan. To accomplish that, you have to first break the existing peptidoglycan that was around the mother cell. Once you execute that break, you have to have new peptidoglycan that is synthesized across the gaps. This will leave a wall band that is very discernible in gram-positives, less so in gram-negatives. In order to then make new peptidoglycan, you have to transport the NAM and NAG units. Remember, those are monosaccharides, so they're quite polar. You have to transport them to the site of growth, which means you have to get them across the cytoplasmic membrane. It's pretty hard to get polar molecules across the cytoplasmic membrane without help. That's accomplished in bacteria with a molecule, it's a lipid molecule called bactoprenol. Bactoprenol will complex with NAM and NAG in the cytoplasm and transport them across the cytoplasmic membrane into the periplasm, where they're then able to be used to form new peptidoglycan. Enzymes called transglycosylases will insert the new NAM-NAG units and catalyze the formation of new glycosidic linkages connecting the carbohydrate portion of peptidoglycan together. This figure shows an overview of what's happening during peptidoglycan synthesis. So to break the existing peptidoglycan, you have to have hydrolysis of the beta-1,4 linkages, that are here, 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 and so on, that are connecting the NAM-NAG units together. That's accomplished by enzymes that are just generically referred to as autolysins. So autolysin activity is what is breaking these beta-1,4 glycosidic linkages connecting together the NAM-NAG units. Bactoprenol, which is shown down here in red, is going to associate in the cytoplasm with NAM-NAG units, and it's going to carry them across the cytoplasmic membrane, okay, and into the periplasmic space, where they're then available for synthesis. As it does that, it's also carrying the whole peptidoglycan base structure, which is actually a NAG-NAM penta, that's a spelling error, pentapeptide unit. So it's the NAM-NAG complexed with a few amino acids. We looked at that structure in a previous lecture. Once you have these units in the periplasmic space, so in here, then you have a set of enzymes called transglycosylases, and they are going to insert those units into the cell wall, into the peptidoglycan. So here we have those structures, they're being inserted into the break point, making new peptidoglycan to close the gap formed where the septum first needs to go. The final step in the synthesis of new peptidoglycan is called transpeptidation. This is the formation of peptide crosslinks or peptide interbridges between adjacent glycan chains, the carbohydrate-based chains, as shown in this figure. So here we have a chain of carbohydrates. Here we have the chain of carbohydrates. They're NAG-NAM units. Here are the amino acids that are part of that base structure, and they are peptide bonded together here, here, and so on. And then you need to connect these together so you can connect these adjacent sheets of peptidoglycan into a much stronger cross-linked structure. That's the act of transpeptidation, bridging those peptides together. This is catalyzed at the division septum by the FTSI protein. Remember, those were one of the proteins that are penicillin binding proteins. So that's why penicillin does its action at the level of the peptide interbridges. So if you have penicillin present, then transpeptidation does not occur. This does not happen, and you're left with peptidoglycan that is not strongly cross-linked. It's therefore weaker, and therefore the cell wall in those organisms would be very susceptible to osmotic stress and very likely to lyse under any kind of osmotic pressure, which is very common in most environments the bacteria are found in, that the osmotic conditions will vary somewhat. So knowing that, you should be able to explain why penicillin is most effective on growing populations of cells. So think about writing out an answer to that using the information in this lecture and in your textbook. That concludes this part of the lecture.